All right, so let's begin today's dissection of the brain. I'm going to remove my jewelry, all right, and set it aside, and then put on my gloves. I have, of course, already got my goggles on. All right, and so what you'll probably have is a specimen stored in a plastic bag like this one, all right. So it is good idea to start. Go ahead, grab your scissors, cut the bag open. Make sure your fingers are out of the way, of course. And for now, we can just set the bag, all right, in the tray. I want to get that out of the way, but I don't want to set it on my workstation, all right? All right, now at this point you could use your paper towels to dry off the specimen. I personally don't find it's necessary. All right, so I want to talk to you about a few things about the brain before we start cutting, okay? Some structures we should see and know right away. Some specimens you get are going to have this opaque white outer covering layer, okay? Those are the meninges, and they can be somewhat difficult to cut through. I want to give you a little insider tip. This is the cerebellum here. There's a particularly thick band through here. If you peel back the cerebellum just ever so slightly, just gently, you can take your scissors, go in and cut through that band, and the whole thing comes off. Easy peasy after that, all right? So this particular specimen does not have the meninges still attached, and that's not a concern. We're going to move on, okay? Something else you should note, in this location we should find the pituitary gland. Now if you have your meninges attached, it's going to be embedded in the meninges. It's a little crunchberry shaped, crunchberry sized object down here embedded in the meninges. When you remove your meninges, you're probably going to tear the pituitary gland off. That's okay, just make sure you look at it before you do and understand where it was and appreciate its size and structure, okay? Now, the company which produced this brain has already removed the meninges, and they've already torn off the pituitary gland, so we're not going to get to see that today. The pituitary gland is, by the way, attached to the hypothalamus by a very thin stalk called the infundibulum. Again, it's a very thin stalk, very easy to tear, and it just rips right away when we remove the meninges, okay? All right, so I'm going to grab my probe so I can point to some structures, all right? Here we see this upside down V shape. If you had your meninges still attached, you would be able to look through them and see the optic nerve. You should see two of them, a right and a left, coming off in this direction. You may even be lucky enough to get a specimen that has some periorbital fat attached to the end of those. Periorbital fat surrounds the eye and protects it from damage. Say if you were to get punched in the head or fall to the ground, it cushions your eye and prevents it from slamming into your eye socket, okay? All right, so you may be fortunate enough to get some of that. Again, we're not because the meninges are removed. Anyway, if you were to remove the meninges, you would see what's left here, this upside down V shape. That's the optic chiasm or chiasma, all right? And that's where part of the optic nerve crosses over, the medial tracts of the optic nerve. They cross over here at the optic chiasma, all right? Up here, we see these white disc shapes, okay? They can be a little difficult to spot on the sheep's brain. Those are the olfactory bulbs. We should be aware of their location, okay? They have more or less the same location on the human brain, all right? All right, so structures we should know from the outside of the brain before we even start cutting. Well, we have the cerebrum here. In the human brain, the cerebrum takes up about 83% of the total volume, total mass of the brain. That's the cerebrum up here. Huge structure in humans, pretty big in sheep as well, at least relative to the rest of the brain. This is a very tiny brain. Back here we have the cerebellum. Cerebellum is Latin for little brain, and it does look like a little brain just kind of hanging off the back, a second little brain back here, all right? We turn it over and we see the brain stem, okay? So we can see the midbrain up top. We see this kind of characteristic bulge of the pons, all right? And down here we have the medulla oblongata, and then we have the spinal cord finally down here, okay? All right. Something else I like to do with the students before we even start cutting into the brain. We've got the cerebrum up here. I've got my thumb on either side of it, and I'm going to peel it apart just ever so slightly. In between, we see this crack. That's known as the longitudinal fissure, the fissure between the right and left hemispheres of the brain. All right, and so if we peel it apart just ever so carefully. Oh, it's tearing, it's tearing. Let's see if we can get it in there. Deep down inside, we can see the corpus callosum, okay? Now, the corpus callosum is a bridge between the right and left hemispheres. If we were to cut this brain straight away, we would have destroyed that, not been able to see it. It is very difficult to see because it will start to tear as soon as you open this up. So if you're unable to see it in the video and you're unable to see it in your dissection at home, just understand that it was there and it is a bridge between the two hemispheres, okay? All right, another structure I like to look at before we even cut into the brain, all right? We put our thumb on the cerebellum and we peel it down just ever so slightly. Be very gentle. A lot of people like to get rough at this point. Just be gentle. Can you see that in there? All right, let's see that. Yeah, you see those 4 bumps? All right, and I want to point them out here. We've got the 2 big bumps up here, the 2 little bumps down below. Can you see those? All right. Those are the superior and the inferior colliculi, okay? Funny word, right? There's a superior pair, they're larger, and there's an inferior pair down below, they are smaller. These are part of the tectum of the midbrain. The tectum, the word means roof of the midbrain, okay? Now, there's another name for these 4 structures. They are collectively known as the corpora quadrigemina, all right? And that means body of 4 twins, or the quadruplets. And a story I like to tell about this is you could easily imagine if you had 4 rowdy boys at home, you could expect to come home and find them sitting on the tectum, or the roof, right? All right, bad joke. So we peel a little further in. There's one more structure I want to show before we even get into the brain, okay? I'm peeling, I've got my thumb this time on the corpora quadrigemina. I'm pulling those 4 down, okay? Can you see in there? Yeah, that little pointy bit right there. All right, that little pointy structure often looks like a diamond depending on your view, okay? That is the pineal body or the pineal gland. All right, remember the pineal body is responsible for converting serotonin to melatonin when it's dark out, which helps prepare the brain for sleep, okay? Now you may have heard of seasonal affective disorder. That's a condition where people get depressed every winter and they also get very sleepy. The reason for that is, we believe, the pineal gland is overactive in these people, converting too much serotonin to melatonin. Too much melatonin makes you very sleepy. Too much serotonin, losing your serotonin makes it difficult to be happy, leads to depression. All right? It's one of those theories that makes total sense on paper. We just need more experiments to prove it, okay? So now I've grabbed my scalpel and I'm ready to begin my first cut. As I explained in the safety portion, I am going to keep my fingers out of the way as I hold the specimen down on the tray, okay? And I'm going to cut from above. Never, ever, ever, never, ever, ever hold it in your hand and cut like this from above. Ever, 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 never do that. All right, so what we're going to do is we're going to make a sagittal section, okay? We're going to cut it right down the middle, okay? And it's easy to do because we're just cutting down the longitudinal fissure, okay? Scalpel's very sharp, so be careful. Try not to destroy your specimen too much. We want a pretty section. We want to see all the structures. And that can be difficult to do, all right? Just go with it, all right? Don't freak yourself out. Just cut very slowly if you need to, right. Wow. This is beautiful. Love the way this specimen looks. Okay, so I'm going to turn it and we can look inside. Probably should have dried it off. It's very wet now. All right, so some structures we can see in the sagittal view here. We have this white strip up here. That is the corpus callosum. That is a strip of white matter that was a bridge between the right and left hemispheres, okay? Up here we can see the cerebrum, all right? And here we can see this oval shape. This oval shape is characteristic of the thalamus. Now, if you remember earlier when we peeked inside, we saw this structure, the pineal gland. If I turn the brain again and we peek inside, you can see it right there, okay? That was the pineal gland, or sometimes called the pineal body, right there. We see the thalamus. Below that is the hypothalamus, okay? A little bit of the optic chiasm is left here, and the infundibulum would have been right around here, connecting to the pituitary gland. Sorry if you couldn't see that. Thalamus, hypothalamus, optic chiasm, pituitary gland would have been down here, connected by the infundibulum. All right, so back here we have the tectum, the superior colliculus here. Now here we have the cerebellum. We also sectioned through the cerebellum. And in here we see this characteristic pattern of white matter. Can you see that? Yeah, it looks kind of like a tree. That structure is called the arbor vitae, or tree of life. Easy to remember. Arbor Day is a day where we plant trees. So, arbor vitae, okay, this little tree pattern of white matter within the gray matter of the cerebellum. All right. Okay, so here is the midbrain. We see the tectum, or roof of the midbrain back here. We see here this characteristic bulge. All right. This characteristic bulge that tells us we are looking at the pons. All right. Okay, here we have the medulla oblongata. Now, I like to take a note here to talk about the word medulla. If you've seen that movie, The Water Boy, there's that section, that scene where Colonel Sanders is yelling at him, "Something's wrong with your medulla oblongata." Ever since then, everybody pronounces this medulla oblongata. But the word medulla shows up in other places throughout the body, and everyone's really careful to say medulla. You can say whatever you want, just understand the history of that word. Okay, the medulla oblongata seen here. We transition into the spinal cord down here. All right, so a couple things we should do at this point. I like to take my probe, all right, and reach in here under the corpus callosum, okay? And I've got a feeling we're gonna find some good stuff in here. We go digging, we go treasure hunting. All right. And we find, oops, here we go. All right. Oh man. There, just the tiniest little sliver of it. Wow. Some of these specimens are pretty poor when it comes to the choroid plexus, which is what I'm digging for. It's a little treasure hunt. Look inside there. We see this little structure, this little bit of tissue here. It's often a dark purplish brown color. Sometimes it can be light pink. Here ours is dark red because this is actually a pretty fresh specimen. I'm really enjoying this brain so far. Unfortunately, it had very little to offer up in terms of choroid plexus. What is the choroid plexus? Well, it's a plexus of highly vascular tissue, which is associated with ependymal cells. The ependymal cells filter blood plasma out of the capillaries there, and they produce cerebrospinal fluid, which they secrete, and then they have little cells on their, little cilia on their apical surface, which swirl and circulate the fluid, okay? So, why do we find choroid plexus in here? Well, this is one of the lateral ventricles. There's a right and left lateral ventricle, okay? This is where we find most of the choroid plexus, all right? And from there, the fluid flows into the third ventricle through the interventricular foramen, which is a little hole, foramina because there are two of them. All right, a little hole between the two ventricles. All right, and from there the fluid will flow down through the cerebral aqueduct into the fourth ventricle. Okay, cerebral aqueduct into the fourth ventricle. Oh, thumbs of the way. All right. All right, so why do we find the choroid plexus here? Well, it's very rich in the lateral ventricles. There's a right and left lateral ventricle, okay? All right, so we find that in the lateral ventricles, it produces that cerebrospinal fluid, which flows through the interventricular foramina into the third ventricle, this space here around the thalamus. It then flows from the third ventricle through the cerebral aqueduct to the fourth ventricle, all right? And from the fourth ventricle, the fourth space here, it flows down into the central canal of the spinal cord. Hopefully you can see that thin little line there that represents the central canal of the spinal cord. Again, flows from the lateral ventricles into the third ventricle, into the fourth ventricle, into the central canal of the spinal cord, okay? Now, why do we have cerebrospinal fluid? Well, it's good for a couple things. It nourishes the brain, okay? We need to do that, all right? It also protects the brain. It serves as a sort of cushioning fluid. Again, if you get punched in the head, your brain's going to bounce around inside your skull. The fluid slows its motion down and cushions some of that shock. Now, the brain's the most important organ. Probably. I mean, you could argue the heart, the liver are also very important, but brain probably most important. We wouldn't be us without it. All right. So the next section we do is a coronal section, and you want to be sure to pick out your ugliest half. What do you think's the ugliest half here, huh? I think this half is uglier. We want to leave the pretty one for us to come back and look at later. So what we're going to do is a coronal section now, where we're going to cut it into front and back portions, anterior and posterior portions, or in the brain, we say rostral and caudal, okay? All right, and I'm going to choose to cut through a section where I'll be cutting through the lateral ventricles, and sure, why not? Let's cut through the third ventricle as well, okay? That scalpel was backwards. All right, we're cutting from above, keeping our fingers free and clear. All right. So now we take the posterior or caudal section of the brain. Oopsie. Now we take the posterior or caudal section of the brain, and we look closely in here. You can see the organization of gray and white matter to the inside, gray matter to the outside. The gray matter to the outside here in the cerebrum is known as the cerebral cortex. The cerebral cortex is where we house lots of anaxonic neurons, which are really important to the functions of the cerebrum. Now, it's gray again because there's going to be little to no myelin here, and white in the white matter because we're going to see bundles of axons there. So a lot of cell bodies, very few axons, no myelin really, little to no myelin. Up here in the gray matter, plenty of myelin down here in the white matter, okay? Now we take this view, and you may have seen a similar view in CT scans if you've ever seen pictures of those. Here we have the lateral ventricle up top and the third ventricle down here. Now on the other side of the brain there would be another lateral ventricle right over here, okay? And we can see those two lateral ventricles, and then down below we would see that third ventricle. In the human scans they tend to be open a little more like that. Now as we turn this, we can actually peek inside, and there is plenty of choroid plexus that I just wasn't able to find, okay? You'll find it mostly in the lateral ventricle, a little bit in the third ventricle, all right? Plenty of that rich pink stuff. All right, can you see that? Rich pink stuff right there, okay? So that about does it for the dissection of the brain. All right, let's move on from here. I'm going to clean up, and then we'll do the dissection of the eye. before you clean up, be sure to take some time to appreciate the structures that you have in front of you. These brains were expensive, you paid for them, and they're a great learning tool. So get as much out of them as you can before you dispose of them, okay? And since we're done with the dissection, feel free to just go ahead and continue doing sections. You can even do slices. Some students like to do that. All right. Give us an appreciation for how the brain looks at different layers, okay? All right. Do whatever you want. What you'll want to do when you're done with any dissection is to get some cleaning solution after you've disposed of everything and washed your tools and put them away. You want to get some cleaning solution. Just go ahead and clean your work surface as well. You don't want to leave this formaldehyde stuff sitting out for kids, pets, or significant others to expose themselves to. You also don't want to get it on yourself.